So, welcome to my thesis defense titled Polyelectrolyte Brush Electrodes for Protein Capture and Release. Let's go. The title of the thesis and its appended papers can be divided into three topics. So the first one is polyelectrolyte brushes in isolation, and the second topic is how polyelectrolyte brushes interact with proteins. And the last topic of the thesis is how polyelectrolyte brushes can be synthesized in a way that makes them compatible with electrochemistry, and in turn, what this can be used for. And this will also be the main outline of the thesis. So I've studied polymer brushes, which are useful for making functional interfaces. Polymer brushes are polymer chains tethered to surfaces at a high graphing density. A free polymer coil in solution will swell to a certain radius, which minimizes its free energy. Polymer brushes are formed when the grafting density of the tether chains becomes so high that the spherical conformation of the polymer is no longer energetically favorable. Instead, the free energy of the polymer brush is minimized by stretching out into a brush height that exceeds the radius of the free polymer coil. So in bioscience, polymer brushes have found two major applications. The first is anti-fouling, which means that molecules are repelled and prevented from reaching the underlying surface. Anti-fouling coatings are useful in implants, drug delivery vesicles, and in biochemical sensors, where one wishes to minimize non-specific surface interactions with proteins and other biomolecules to prevent undesired effects. The second application is immobilization of a specific kind of molecule. For instance, polymer brushes can be used to immobilize and store enzymes on a surface to perform biocatalysis in the production of food and pharmaceuticals, for instance. It can also be used to promote adhesion of cells to surfaces. Or it could be used to bind receptor molecules for use in biosensing. So many researchers study brushes, but they typically only do so with one or the other property and in isolation. A polymer brush that could conveniently and reversibly switch its properties between these two functions on demand would be highly appealing. However, despite the appeal, such an interface has not yet been developed. In this thesis, I studied charged polymer brushes called polyelectrolyte brushes. I specifically studied pH-responsive brushes, one example of which is polymethacrylic acid, shown here. pH response means that PMAA changes between a negative charge state, that is deprotonated, and a neutral protonated state, depending on the pH of its environment. The relationship that defines this proton association-dissociation relationship, as you might have heard, is called the pKa. pH-responsive brushes are highly suitable for making interfaces that switch. By setting the pH, you can tune attractive and repulsive interactions between other charged objects like proteins. This is a highly popular and perhaps most used application of polyelectrolyte brushes. However, it's also a phenomenon which is poorly understood because there are many parameters that affect electrostatic interactions, and experimental outcomes can be difficult to interpret and to model. So in the first publication, I studied and learned polymerization of polyelectrolyte brushes. To produce them, I used a method called ATRP, which in the scheme here to the left, I show you a representative reaction protocol that I use to produce so-called PDA brushes, which contains a tertiary amine functional group that can become positively charged. Using a highly similar protocol, I also synthesized anionic negatively charged polyelectrolyte brushes. The brushes were performed by using grafting from polymerization technique. This means that we bound polymerization initiators to the surface and grew the polymers directly off of the surface. Producing polyelectrolyte brushes this way results in a very high grafting density, promoting highly stretched out polymers. With this method, we get a controlled, even rate of polymer growth, which results in an even, well-defined nanoscale polymer layer. A central characterization technique used by me in this thesis is called surface plasmon resonance. It's an optical sensing method that can determine the refractive index when it changes on thin metal surfaces. So you produce the plasmon resonance by irradiating the thin metal gold film, and this is what you see in the spectra to the right here. And when the plasmon angle shifts, this is because you've increased the refractive index of the surface, for instance, by putting a brush on it. The shift in the spectra can be used to determine the brush thickness if we know the refractive index of this layer. So in air, this is easy to do. However, in solution, this is difficult because brushes hydrate, which makes the refractive index unknown, preventing an accurate determination of the brush height. One effective solution to this problem is to combine SPR with a macromolecular probe, this polymer in red here, which does not bind to the brush. This allows us to measure two spectra, one without and one with, and this gives us enough information to simultaneously determine both the refractive index and the thickness of the brush. This has been used by our research group and collaborators in the past to determine brush heights of neutral brushes like polyethylene glycol and Pnyta. Crucial for making this work also for polyelectrolyte brushes was finding a probe molecule which is non-interacting with a polyelectrolyte brush, both when it is charged and when it is neutral. We found that by using polyethylene glycol, we could achieve this and determine, for instance, for this polymer PDA, poly-2-ethyl diethyl methacrylate, we could determine the charged brush height to be 118 nanometer and the collapsed brush height to be 63 nanometer. To summarize the results of paper 1, we performed ATRP to produce different kinds of polyelectrolyte brushes. We characterized the brush heights using the non-interacting probe method. And crucial for achieving this for polyelectrolyte brushes was using synthetic polymers, such as PEG, as the macromolecular probe. Proteins, which are otherwise used as probes for neutral brushes, were found to be unsuitable since they significantly interacted with the polyelectrolyte brush, preventing height determination. So having learned how to make polyelectrolyte brushes, I next used them to study enzyme immobilization. So there are several options for the chemical interactions which can be used to attach the enzymes to surfaces, as I show here to the left. But just binding enzymes to a surface is not enough. For it to be practical, it should display a high capacity for irreversible immobilization, yet it should be gentle enough so that the enzymes actually retain their function while on the surface. We chose to study electrostatic, obviously, and covalent immobilization to polyelectrolyte brushes, since we knew from literature that brushes can immobilize large quantities of enzymes for efficient biocatalysis by these methods. However, the specific gain of placing them in brushes had never been systematically investigated with placing them on a flat surface and compare this with the brush case. But still using the same functional groups that are present in the brush and on the surface. First, we wanted to compare the surface coverage. So we used to test a model enzyme, glucose oxidase, and immobilized it to these surfaces. And we quantified the amount of enzyme per surface area in the brushes compared to the self-assembled monolayers. So here are the three cases for the brushes we tested, and here are the self-assembled monolayers. What we see is that the brushes clearly store a larger quantity of enzymes in multiple layers, while the self-assembled monolayers can at most produce a single layer of enzyme on the surface. We then measured the activity as a function of the bound quantity of enzymes. We observed that a high degree of activity retention is present for in polyelectrolyte brushes and that the self-assembled monolayers had very low activity in comparison. In fact, we fully retained the enzyme activity compared to the bulk enzyme solution activity. The reason for the very high retention of activity in polyelectrolyte brushes is that they provide a soft, hydrated, three-dimensional support scaffold. This allows for high accessibility of the reactants and the products to diffuse to and from the active site of the enzymes. In summary of paper 3, we found that polyelectrolyte brushes are superior in terms of surface coverage quantity and in the retention of enzyme activity. In fact, the activity of enzymes in brushes could be fully preserved. So in this paper, paper 3, enzymes were immobilized to brushes using conventional electrostatic interactions and covalent functionalization. So these are great for placing and keeping enzymes in brushes permanently for performing biocatalysis. However, these approaches are not so good if the scope is to only temporarily capture and then subsequently release the bound proteins later. Now, we make a transition from the established methods into a new method for protein immobilization. So in paper 1, I discovered an attractive interaction between proteins and polyacidic brushes like polyacrylic acid and polymethacrylic acid under acidic conditions. At low pH, these brushes are neutral but protonated, enabling them to hydrogen bond with molecules that they are exposed to through the carboxylic acid group. PMAA is known to form hydrogen bond complexes with other synthetic polymers in solution. And in this case, PMAA immobilizes significant quantities of protein at a relatively high value of pH 5. To test the protein binding capacity of PMAA, first, an SPR sensor was exposed to a protein solution. Secondly, the surface was rinsed and dried, and we measured in SPR the air scan to produce a surface coverage quantification of that protein. And third, the pH was raised to a value that exceeds both the pKa and the isoelectric point, the pi, of the protein, which breaks the hydrogen bonds and induces a net electrostatic repulsion between the protein and the brush, completely regenerating the brush. Then this surface can be reused to perform another experiment and another, and you can carry on like this. And we obtained a very high binding capacity of many different proteins in the range of micrograms per square centimeter, making this a generic method to capture large quantities of any protein to surfaces. Importantly, the interaction was confirmed to be non-electrostatic by the lack of correlation between the protein pi and the amount of surface coverage. Previously, we showed that enzymes could retain their activity when immobilized to polyelectrolyte brush by electrostatic attraction. What about now? Because now we immobilize proteins to a neutral brush, we need to check that this also preserves the structure and the function of the proteins. So to do so, we used circular dichromosome spectroscopy, which detects the secondary structure of proteins. We used this to verify that when BSA is desorbed from PMAA at pH 8, the spectra looks almost identical to the stock solution pH of BSA, confirming that the structure is preserved. Similarly, we check that glucose oxidase retains its enzymatic activity before and after capture and release. Of course, more proteins need to be tested and specific cases need to be evaluated. But it highlights that the method is not invasive to water-soluble proteins like BSA and GOX. In conclusion of paper 4, we can state that polyacidic brushes in neutral state has a large capacity to bind proteins by non-electrostatic interactions. This is a new method for protein immobilization that is distinctly different compared to conventional immobilization by electrostatic attraction. Importantly, complete release of proteins was achieved, enabling multiple capture and release cycles without fouling. The use of polyelectrolyte brushes in this way makes them highly appealing for applications such as protein purification. With this, I'd like to return for a second to just discussing polyelectrolytes, because we realized the importance of the neutral state of brushes, and this made us very interested in the pivot point, the pKa, between the charged and the neutral state of the polymer brushes. The parameter that we were the most interested in studying was the total salt concentration, as we noticed that there is a large uncertainty in literature of how it affects the pKa. We chose to study these three polyelectrolytes. We used SPR, since this is an ideal tool to study the pKa, because it detects the refractive index associated with going between the charged and the neutral state of the brush. As this graph shows, SPR detects the switch from charged to neutral, allowing detection of the pKa at 1 millimolar salt concentration. Then we systematically varied the salt concentration and recorded the SPR shift as a function of the pH of the buffer solution. For each increase in the total salt concentration, the buffer pKa shifts to higher values. By examining the pKa of both anionic and cationic brushes, we see that this is a general feature of pH responsive brushes. For anionic brushes, the pKa decreases with increasing salt concentration, and the opposite trend is observed for the cationic brush. And the total change in pKa between 1 and 510 millimolar salt concentration amounts to almost 3 units, which is a remarkable change in the pKa compared to the corresponding case for polyelectrolytes when they are free in solution. However, it's not enough to do this just by SPR. We need to evaluate this and verify it using other techniques as well. And for this reason, we used QCMD, which is another surface-sensitive technique that is highly complementary to SPR. It also detects the degree of charging, but by a different mechanism. And the QCM results indeed verified the SPR results that we see here. And that was still not enough, so we performed an IR study as well. So here you see IR spectra to the left, which is a direct measurement of the protonation state of, in this case, polyacylic acid. We use this to, again, generate curves showing the degree of charging. And using a third technique, we could show, again, that the total salt concentration shifts the pKa by several units. We wanted to study and know what the implications of these shifts in the pKa as function of the salt concentration has for protein immobilization. So to do this, we studied BSA immobilization to PMAA, first at physiological salt concentration, 150 millimolar, and for 3 different pHs, 5, 6, and 7. So we know from paper 4 that proteins will immobilize to BSA when the PMAA brush is neutral at pH 5. This is what we see here. But that it will be repelled from the brush when it is predominantly charged at 6 and 7. What happens now if we do this at a lower salt concentration? Now BSA immobilizes at all 3 pHs, 5, 6, and 7. And our explanation is that at 1 millimolar total salt concentration, PMAA stays predominantly neutral even up to pH 7, which continues to then give rise to protein immobilization as you increase the pH. So by changing the total salt concentration, we can trigger protein binding through non-electrostatic interactions at even higher pH than 5. Furthermore, our results provide a new explanation to a phenomenon called immobilization of the wrong side of the PI. This is a phenomenon observed at low salt concentration, where proteins immobilize the brushes despite what is thought to be a net electrostatic repulsion. Our characterization of the brush pKa at low salt concentration shows that the surface is not actually negative at these pH values, and that immobilization occurs by non-electrostatic interactions. We also confirmed that the same effect occurs also for positively charged brushes like PDR. So the salt concentration of polyelectrolyte brushes shifts the pKa by several units, which is larger than the corresponding case for free polyelectrolytes in solution. Consequently, extreme pH values are required to charge brushes at low salt concentration. And therefore, protein immobilization at low salt concentration is likely driven by neutral intermolecular interactions like hydrogen bonding. The effects we witness here are relevant not only for polyelectrolyte brush interfaces, but for any pH-responsive polymer environment with a sufficiently high density of acidic or basic functional groups. Until now, the pH was switched by pumping a new buffer liquid, which is a slow and practical method. Electrochemistry offers a rapid, non-invasive method for pH switching because it only switches the pH of the local environment surrounding the brush. But to achieve this, we needed an electrochemically stable linker between the surface and the brush. Thiol surface initiators, which are used up to this point, could not be used since the thiol gold bond is electrochemically unstable and it degrafts even when you apply modest, weak electrochemical potentials. Instead, we used diazonium salts to produce these linkers, as shown here, which permitted repeated switching without degradation. We also needed a redox reaction that produces pH gradients by applying an electrochemical signal. And this is an example of a very useful reaction where proteins are consumed at the electrode surface together with oxygen to produce either water or hydrogen peroxide. By performing CV scans between 0 and -1 volt, we detected an electrochemical process at low potentials here, which showed a very strong pH dependence. And we verified that it was indeed these reactions by also purging the solution from oxygen, upon which the process disappeared. We could prove that the proton consumption at the interface switched the polymethacrylic acid brush by using in situ electrochemical QCMD. So in this graph to the right, we show a rectangular shape of the signals, which indicate that we are reversibly switching the brush between a neutral state, a charged state, and back and forth like this. The crosses in the graph represent the experiment where the brush was switched using electrochemistry by applying -1 volt. And we did that in the blue shaded regions of the graph. Compared to the pump experiment with the circles, we see that the response is nearly identical, except that the pump is much slower in switching the pH. And this is because it requires a complete replacement of the flow cell liquid to switch the pH, something which the electrochemical method does not require. It only requires switching the pH at the very vicinity of the electrode surface, making it much faster. So in paper 4, we released proteins from polymethacrylic acid brushes by exposing the liquid to a high pH solution. Now we no longer need to do that. Now we can, by the click of the computer button, electronically release proteins from the surface. And we can prove that this is the case by another in situ electrochemical QCMD experiment, where we begin by loading proteins at the start of the experiment at pH 5. Once the proteins are loaded to the surface, we then move on to tunably release the proteins that are bound by applying cyclic voltammetry scans to -1 volt. And for each scan, a dose of protein is released from the surface until enough scans have been repeated such that the signal returns to the baseline and we can restart the process by loading more proteins to the surface. So with this electrochemical reaction that we are using here, we can only increase the pH, so turn off the protein interaction. The question is, can we find a way to operate these electrodes so that we can turn on the protein interactions in a pH 7 liquid, a neutral buffered liquid? And yes, we can. Luckily, there are more redox reactions which can be electrochemically activated. For instance, hydroquinone, which allowed switching the pH from neutral pH 7.4 to acidic conditions. And yet again, another in situ electrochemical QCMD measurement where we go now from pH 7.4 to 5, and we reversibly switch it back and forth, again confirming it with just manually changing the pH. By activating the surface to become acidic, this allows us not only to capture proteins out of buffered liquid to collect the sample, for instance, it also crucially permits a surface to hold a large quantity of protein for on-demand release into a buffered liquid with minimal interference to the system. So we could achieve this not only using hydroquinone, but we could also use ascorbic acid, dopamine, and even glucose to produce these pH gradients. To summarize paper 5, reversible electrochemical switching of polyelectrolyte brushes enable us to build a bioelectrode which can capture proteins by hydrogen bonds, release proteins by electrostatic repulsion, and this design was enabled by insights from all 4 of the first 4 papers of the thesis into how proteins and polyelectrolyte brushes interact as a function of pH. And secondly, making use of the underlying surface as an electrode to produce local, non-invasive pH gradients. Reconnecting to the introduction, we have created a polymer brush interface which can interchangeably and reversibly switch between a capture and a non-fouling state. This is interesting not only from a scientific point of view, because there is a commercial demand in the biotechnology industry for developing materials that can accomplish these tasks. So first, the capture of proteins on an industrial production scale to purify them. And secondly, for analytical applications to collect samples of proteins. The second reason is we need to deliver biomolecules on demand, and this is a convenient method of creating a bioelectrode that can be interfaced with tissue and living cells. So this is today very relevant, since many of the best drugs that we know for treating the most challenging diseases are biological molecules. They are protein, oligonucleotides, carbohydrates, and liposomes, which are widely used as pharmaceuticals. And despite being very successful, big challenges remain to be solved in improving and reducing the cost of the biotechnology behind these life-saving treatments. My hope is that the thesis studies here has resulted in new tools and understanding which could play some role in addressing these challenges. So to wrap up, I'd like to make some acknowledgements, of course, to my wonderful research group led by Andreas, who is my supervisor, to the current and the former members, to our collaborators, and to all of the people at Applied Surface Chemistry, and notably Annette and Anders, my assistant supervisor and examiner, and of course, all of the other helpful and nice colleagues at Chalmers. And also, of course, the funding sources. Thank you very much.